Hi friends, welcome back to KPS Bio Tutorials. This is the 9th episode of Immunology lecture series. In the last few episodes, we have been talking about the cells of immune system, in which we already covered about the cells of the lymphoid lineage. We discussed about cells in the lymphoid lineage, that is the NK cells, T cells and B cells. And in today's episode, I would like to talk about the cells of myeloid lineage. So let us go to the topic. Once again, welcome back to today's topic, that is cells of myeloid lineage. The cells like macrophages, dendritic cells, eosinophils, neutrophil, basophil, mast cells, etc. are belonging to the cells of myeloid lineage. During the hematopoiesis, from the multipotent hematopoietic stem cells or hemocytoblasts, two categories of cells were derived. One is called myeloid stem cell, other one lymphoid stem cell. From the myeloid stem cells, megakaryoblast, proerythroblast, myeloblast and monoblast cells were later derived. From these myeloblast cells, the basophil, neutrophil, eosinophil, and from the monoblast cells, monocytes, and from the monocytes, the macrophages were derived. This is the flowchart of the hematopoiesis. Now let us examine about the mononuclear phagocytes. The mononuclear phagocytes are of two categories: a monocytes and macrophages and dendritic cells. Monocytes are mononuclear phagocytes which circulate within the blood, whereas macrophages and dendritic cells are present in the tissues. During the hematopoiesis in the bone marrow, granulocyte monocyte progenitor cells differentiate into a pro-monocytes. Here you can see this is a granulocyte monocyte progenitor cell, which may differentiate into a cell namely monoblast, or which is the pro-monocytes, which will leave the bone marrow and enter the blood, where they further differentiate into mature monocytes. And these monocytes circulate in the bloodstream for about 8 hours, during which they enlarge. They then migrate into the tissues and differentiate into specific tissue macrophage or into dendritic cells. So from the pro-monocytes, the monocyte will develop. Monocyte will be present in the blood for 8 hours. After that, it migrates into tissues, and in the tissues it differentiates into macrophages or dendritic cells. That is the course of development of monocytes and macrophages or dendritic cells. Now let us examine what are the changes involving in differentiation of monocytes into a tissue macrophage. The cell enlarges 5 to 10 fold. The monocyte cells, as they enter the tissues, it enlarges 5 to 10 fold. Here you can see a monocyte will be enlarged 5 to 10 fold to form macrophage, and its intracellular organelles increase in both number and complexity. The number of intracellular organelles like mitochondria, Golgi bodies, ribosomes, lysosomes, etc. will be enlarged, as well as their number is increased. This is the second most important change occurring in the monocytes while they are transforming into macrophages. And it acquires increased phagocytic ability and produces higher level of hydrolytic enzymes and being to secrete a variety of soluble factors. Its total phagocytic ability will be increased by the increased production of hydrolytic enzymes, and also it secretes a variety of soluble factors in order to engulf or in order to phagocytosis the foreign pathogens. So these are the major changes occurring in a monocyte while they are transforming into macrophage. Their cell will be enlarged 5 to 10 folds, and the cellular organelles are increased in number and complexity and increased phagocytic ability. Then these macrophages are dispersed throughout the body. Some take up residence in particular tissues, fixed macrophages. They are called fixed macrophages, whereas other remain motile. There are two types of macrophages based on their mobility. Some are fixed in certain tissues, they are called fixed macrophages, and some others are free or wandering type. They may be able to move or wander in different areas of our body. The free macrophages travel by amoeboid movements throughout the tissues. In different tissues, it can move by amoeboid movements. Then macrophages serve different functions in different tissues. They are named as according to their tissue location. That means they are fixed macrophages. They are fixed or they are present solely in particular tissues. According to the tissues in which they reside, they are named. First of all, the mesanglial cells, they are present in kidney. Mesanglial cells are the macrophages present in the kidney, whereas microglial cells, they are present in brain, osteoclasts present in bone, alveolar macrophages present in lungs, histiocytes present in connective tissues, Kupffer cells present in liver. So tissue macrophages are of different types. This classification is based on their location or the tissues in which they are residing. Then how do macrophages work? First of all, we can discuss what is the major function of a macrophage. They ingest and digest exogenous antigens such as whole microorganisms and insoluble particles, and endogenous matters such as injured or dead host cells, cellular debris, and activated clotting factors. So this is the major function of macrophage. They ingest and digest exogenous antigens. That means the pathogenic microorganisms like viruses, bacteria, etc. And it also works on endogenous matters like injured cells, injured or dead host cells, cellular debris, and activated clotting factors. That means there are debris from the normal cell cycle. As a result, they are formed as a result of normal cell cycle. Certain cells will be destroyed, and the debris of such damaged or dead host cells will be ingested and digested by these macrophages. Then let us examine what are the steps of phagocytosis. The first step in phagocytosis is chemotaxis. As the name indicates, it is chemotaxis. That means attracting substances or cells with the help of certain chemicals. That is called chemotaxis. Macrophages are attracted by and move towards a variety of substances generated in an immune response. As a result of a molecule generated in immune response, macrophages will be attracted towards such cells. Then second one is adherence. Adherence of antigen to a macrophage cell membrane. Antigen will be adhered or it come in close contact with the macrophage The complex antigens such as whole bacterial cells or viral particles tend to adhere well and are readily phagocytosed. The complex antigens like bacterial or viral particles tend to adhere well and readily phagocytosed. Isolated proteins and encapsulated bacteria tend to adhere poorly and are less readily phagocytosed. That means the larger viral as well as bacterial cells, viral particles and bacterial cells are very easily adhered to the adhered to the macrophages, and they are easily phagocytosed. Whereas small protein particles, they adhere poorly and less readily phagocytosed. Then the third step of phagocytosis is the development of pseudopodia. The adherence of the antigen to the macrophage induces the membrane protrusions called pseudopodia to extend around the attached material. So when an antigen is attached to the macrophage, a membrane protrusion will be developed around the antigen and it help to engulf the antigen by the phagocytes. Then fusion of the pseudopodia. After developing the pseudopodia around the antigen attached to the phagocytes, then these pseudopodia fuse so that the antigen will be internalized by the macrophage and it will be captured in a structure namely phagosome. It is a membrane bound structure. So now the antigen is get engulfed by the phagocyte or macrophage and it is enveloped by a membrane bound structure called phagosome. Then the fifth step of phagocytosis, endocytic processing. A phagosome then moves towards the cell interior where it fuses with lysosome to form a phagolysosome. The phagosome with the antigen. Phagosome is actually a packed antigen. This packed antigen then fuses with the lysosome present inside the macrophage to form a structure namely phagolysosome. The next step is the digestion. The lysosome contains lysozyme and a variety of other hydrolytic enzymes that digest the ingested material. Lysosome, the lysosomes containing multiple types of digestive enzyme, hydrolytic enzymes. These enzymes work on the antigen within the phagolysosome and they are get digested. Then exocytosis. The digested contents of the phagolysosome are then eliminated in a process called exocytosis. After digestion or destruction of the antigen, it is the debris or the digested contents will be eliminated from the macrophage by a process called exocytosis. That means actually a reversal of the endocytosis. Then the last step is antigen presentation. A peptide derived by the antigen processing is presented on class 2 MHC molecules. After digestion of the antigens, as a result of this digestion, some peptide molecules will be derived from the antigen, and that peptide indicate the presence of the antigen which will be presented on class 2 MHC molecule. This process is called antigen presentation and it is for the identification by other cells like T helper cells. The T helper cells can recognize the antigen presented on class 2 MHC molecules. and it will elicit the cell-mediated immunity. So this is about the whole reactions or whole steps of phagocytosis by a macrophage. Then let us look at a term, namely opsonization. The macrophage membrane has receptors for certain classes of antibody. If an antigen, for example bacterium, is coated with an appropriate antibody, the complex of antigen and antibody bind to antibody receptors on macrophage membrane more readily than antigen alone, and phagocytosis is enhanced. Here, a macrophage membrane, when we examine the membrane of the macrophage, you can see there are a number of receptors, namely Fc receptors. On these receptors, the Fc region of antibodies can bind. Then, when a pathogen, for example bacterium, when an antigen like bacterium, it may be already coated with an antibody. So an antibody bound antigen come close with the macrophage. In the macrophage there is already a receptor for the Fc region of the antibody. So the Fc region of the antibody binds on these receptors, Fc receptors on the macrophage. Thereby these antigens are very closely and readily adhere to the macrophage with the help of this antibody. So actually the presence of this antibody enhances the effectiveness of phagocytosis. The macrophage, even in the absence of these antibodies, the macrophage can endocytosis these antigens. But if antibody is there, it will make the process much easier than in the absence of the antibody. So here antibodies bound to the antigens are acting as a mediator between the macrophage and the antigen, and it enhances the process of phagocytosis. This enhancing of the phagocytosis with the help of the antibody and antibody receptor interaction is called opsonization. Here the mediator antibody is termed as an opsonin. It works as an opsonin, and the entire process is said to be opsonization. In one study, for example, the rate of phagocytosis of an antigen was 4,000-fold higher in presence of a specific antibody to antigen than in its absence. In one study, the presence of antibody will increase the rate of phagocytosis by 4,000-fold. So that is the importance of the opsonization. Thereby the antibody functions as a opsonin. That means a molecule that binds to both antigen and macrophage and enhance the phagocytosis. Any molecule which mediates the reaction between antigen and the macrophage and it will enhance the rate of phagocytosis is said to be opsonin. So the process by which particulate antigens are rendered more susceptible to phagocytosis is called opsonization. This entire process is said to be opsonization. Then let us discuss about the another cells in the myeloid lineage, that is granulocytic cells. You know, in our blood there are many granulocytes. The granulocytes are classified as neutrophils, eosinophils and basophils. This classification is based on the cellular morphology and cytoplasmic staining characteristics. Cellular morphology, that means the shape and structure of the cell, as well as the staining properties of the cell. This classification is made. First of all, we discuss about neutrophils. You are very familiar with neutrophils. The multi-lobed nucleus, its major peculiarity is the multi-lobed nucleus and granulated granulated cytoplasm that stain with both acidic and basic dyes. It can be stained with both acidic and basic stains, thereby it is called neutrophils. It is often called a polymorphonuclear leukocyte or PMN for its multi-lobed nucleus, since it is having a multi-lobed 1, 2, 3, sometimes 3 lobes in their nucleus. Thereby they are called polymorphonuclear leukocytes or PMN. These neutrophils are produced by hematopoiesis in bone marrow, as we discussed earlier. As a result of the hematopoiesis, the neutrophils are developed from the myeloblast cells, and they are released into the peripheral blood and circulate for 7 to 10 hours before migrating into the tissues. So just like in the case of monocyte, as we already discussed, these neutrophils are also released into peripheral blood and circulate in the blood for 7 to 10 hours. Then it may migrate into the tissues. The migration of neutrophil into the tissues is said to be extravasation, where they have a lifespan of only a few days. Within the tissues, it may live only for a few days. And another important feature about neutrophil is these are the cells first to arrive at the site of inflammation. If there is an inflammation, the first cell which arrives at the site of inflammation is neutrophil. The resulting transient increase in the number of circulating neutrophil is called leukocytosis. So, if there is any inflammation at that region, there will be a rapid increase, a transient increase in the number of circulating neutrophil. That condition is said to be leukocytosis. This is used as a medical indication of infection. So, if there is leukocytosis, that indicates there is an infection. Now, let us examine eosinophils. Eosinophils, like neutrophils, they are motile phagocytic cells that can migrate from blood into tissue spaces. Just like neutrophil, eosinophils also able to migrate to tissues from blood. But their phagocytic role is significantly less important when compared with neutrophils. And it is thought that they play a role in the defense against parasitic organisms. The secreted contents of eosinophilic granules may damage the parasite membrane. So they are having certain granules which contain certain substances which may destroy or damage the parasitic membrane. So eosinophils are mainly referred against parasitic organisms, and they are said to be eosinophil because they are stained by acidic stains. Then basophils are non-phagocytic granulocytes that function by releasing pharmacologically active substances from their cytoplasmic granules. These substances play a major role in certain allergic responses. So the basophils, as the name is derived from the term base, that means they are stained with the basic stains. And they are non-phagocytic granulocytes. They have granules, but non-phagocytic. Their granules contain pharmacologically active substances like histamine, which mediate the allergic responses. The next cell to discuss is mast cells. These are also formed in bone marrow by hematopoiesis. They are formed from the mast cell precursors, and they are released into the blood as undifferentiated cells. They do not differentiate until they leave the blood and enter the tissues. They leave the blood, then enter the tissues. Within the tissues, they are developed into a mast cell. These mast cells can be found in a wide variety of tissues, including skin, connective tissues of various organs, and mucosal epithelial tissue of respiratory, genitourinary, and digestive tracts. Like circulating basophils, these cells have large number of cytoplasmic granules and contain histamine and other pharmacologically active substances. These cells contain number of granules with pharmacologically active substances like histamine. And mast cells, together with blood basophils, play an important role in the development of allergies. So these mast cell and basophils are working hand in hand to develop allergic reactions. Here on the membrane of the mast cell, here you can see certain receptors, namely Fc receptors, specifically for IgE antibody. You know, this IgE antibody is mediating the allergic reactions because the IgE antibody binds with allergens, and when an allergen binds with the IgE binds on the mast cell with the help of these Fc receptors on the mast cells, and thereby this reaction mediates the releasing or degranulation of pharmacologically active substances within the mast cell. These pharmacologically active substances cause the allergic reactions. Now the last cell we have to discuss is the dendritic cells. As the name indicates, they are derived. The term dendritic cell is derived from the term dendron because they look like dendrons and dendrites of the neuron because they have branched extensions to their cell membranes. So they have long membrane extensions that resemble the dendritics of a nerve cell. They are called dendritic cells or DC. Their major function is the presentation of antigen to T helper cells. They are most potent antigen presenting cells. So their major role is presentation of antigens to the T helper cells. There are 4 types of dendritic cells are known. One is Langerhans cells. You know, these dendritic cells are actually derived from the monocytes. A macrophage also derived from the monocytes, and there are different types, namely Langerhans cells, interstitial cells, myeloid dendritic cells, and lymphoid dendritic cells. Langerhans cells are present on our skin, interstitial cells present in the interstitium, myeloid dendritic cells are derived from the myeloid cells, and lymphoid dendritic cells, they are present in the lymphoid organs. And each arises from hematopoietic stem cells. You know, all these cells are derived from hematopoietic stem cells. And the most important thing about the dendritic cells are they are constitutively express high level of both class 2 MHC molecule and members of the co-stimulatory B7 family. These class 2 MHC molecule and the B7 molecules are required for the antigen presentation. So these 2 molecules will be constitutively expressed on the dendritic cells. So this feature make them as a most potent antigen presenting cells. So they are most potent antigen presenting cells than macrophages and B cells. There is another type of dendritic cell, namely follicular dendritic cell, which is entirely different from the dendritic cell that we now discussed. Its function is entirely different from the antigen presenting cells. They are not antigen presenting cells because they do not express class 2 MHC molecule and therefore not an antigen presenting cell for T helper cells. These dendritic cells were named for their exclusive location in lymph follicle. They are located in the lymph follicle, thereby they are called follicular dendritic cells. They have an entirely different function which will be discussed in the coming episodes. So this is about an overview of the tissue, the cells of myeloid lineage. Thereby, I think we discussed about all types of immune cells. I hope you understood the topic very well, and I will be coming back with the remaining chapters in the following episodes. If you understood the topics very well, you like the video and also share with your friends. If you are not yet subscribed to this channel, please subscribe. Also make your comments in the comment box. So we will meet again in the next episode. Till then, goodbye to all of you.